Good afternoon, dear students. I am glad to welcome you to the 12th practical lesson, which is devoted to the study of linked inheritance and analysis of gene coupling. The lesson will be conducted by me, Andrei Fedorovich, assistant of the Department of General and Molecular Biology. This slide shows the questions. I'll write them down in the report. I want you to meet Thomas Morgan. What do you think? Who is it? Thomas Morgan is American biologist and geneticist. One of the founders of genetics, laureate Nobel Prize in physiology or medicine, mogenist, experimentally proved the chromosomal theory of heredity. Each gene occupies a specific place in the chromosome locus. The genes in the chromosome are arranged linearly in a certain sequence. Different chromosomes contain different numbers of genes. The set of genes on each non-homologous chromosome in a unique. The genes of a single chromosome from a linkage group and are inherited together, so linked. The number of linkage groups is equal to the number of chromosomes in the haploid set. For example, Drosophila has 4, has 20, allelic genes can be exchanged between homologous chromosomes, so crossing over. The frequency of crossing is directly proportional to the distance between genes in the coupling group. For the unit of distance between genes in the coupling group, a special unit is taken, mogenate. So 1 mogenate is 1% of the crossing over. 4 and 5 paragraphs are reflected in the Morgan art. Thomas Morgan's law. The genes of a single chromosome from a linkage group and inherited together, though linked. The number of linkage groups is equal to the number of chromosomes in the haploid set. In experiments on Drosophila, it was found that the development of traits that are inherited concatenated, so controlled by the genes of a single chromosome. The genes for body color and wing length are localized in a single pair of homologous chromosomes. B, gray body color gene, small b, black body color gene, big V, normal wings, long wings gene, small v, gene for short wings or rudimentary wings. Crossing gray flies with normal wings and black flies with rudimentary wings will give the first generation of gray hybrids with normal wings. During the analysis crossing, males were selected from F first. Science, it is known that male Drosophila have achiasmatic spermatogenesis. So crossing does not occur and the completeness of gene coupling is not violated. As a result of this crossing, of two phenotypes are born similar to the original parent forms and in equal quantities, splitting according to the one-to-one phenotype. Considered together, the results of both crosses suggest that the development of the analyzed traits is controlled by different genes, and like inheritance is explained by the localization of genes in one chromosome. The completeness of the coupling in this case isn't violated. This gene coupling is complete. Females were selected from F first to study incomplete coupling. In females, crossing occurs during gametogenesis, so heterozygote individual form an additional varieties of crossover gametes. The probability of their formation is determined by the probability of crossing over. So it depends on the distance between the genes in the coupling group. In this example, splitting by phenotype in the offsprings is obtained as follows. Gray flies with long wings, 41 and 5%. Black with short wings, 41 and 5%. Gray Brachypterius, 8 and 5%. The long winged black 8 and 5%. Thus, the probability of the appearance of recombinant individuals in the offspring is 17%. Therefore, the distance between the B and V genes in the coupling group is 17 mogenate. The predominance of gray long-winged and black short-winged flies in the offspring indicates the B and V genes. B and V are indeed linked. One of the other hand, the appearance of recombinant individuals suggests that in a certain number of cases there is a break in the coupling between the B and M genes and the B and M small genes. This is the result of a crossing over. An example of complete gene coupling in humans is the inheritance of the Rh factor. It is caused by three pairs of C, D, and K that are closely linked to each other, so the inheritance of RH belonging occurs according to the type of monohybrid crossing. Another example of a close coupling of genes in humans is the inheritance of cataracts and polydactyly. The genes of hemophilia and color blindness are localized in the X chromosome, at the distance of 9 and 8 mongenites. So they are subject to crossing over, so they are inherited as incompletely linked. Autosomal Rh factor genes and forms of red blood cells located at a distance of 3 mogenate from each other and is also an example of incompleted coupling. Excuse me. In 1909, Jensen, while studying meiosis in amphibians, found chiasms, crosses of chromosomes, which are cytological evidence of crossing over. Since that time, many attempts have been made to explain the mechanism of this phenomenon. There are several theories of crossing over. Two hypotheses are the most common. First hypothesis, the hypothesis of divide and reunification. Second, the hypothesis of selective copying. This first hypothesis of divide and reunification. According to this hypothesis, crossing over is performed by breaks occurring simultaneously at identical points of two parent DNA molecules and subsequent covalent joining of the formed fragments in new combinations. According to scientist Halliday, the break occurs in DNA strands of the same polarity. Whitehouse and Hastings believe that homologous chromosomes break strands of DNA not the same but different polarities. Despite different points of view, it is common to believe that crossing over begins with single strand breaks. Of great importance for the implementation of crossing over are the following enzymes: endonuclease causing a break in the DNA strand, exonuclease expanding breaks, DNA polymerase filling the gaps with nucleotides, ligase finally reuniting the broken ends of the DNA strands. A lack of one of the enzymes can lead to violation of the ability to recombine. This crossing over occurs without DNA replication. and the crossover DNA molecule is made up of the substance of two parent molecules. That second hypothesis of selective coping. When crossing over, there is no exchange of sites between the parent DNA molecules, but the genetic information recorded in them is partially transmitted to the newly synthesized molecule during replication. This molecule, when growing first, uses the thread of one of the parent molecules as a matrix, and then switches to using the thread of the other parent molecule. Thus, crossing over occurs during DNA replication. The connection of the crossover DNA molecule with the parent of purely informational. Part of the information received is supplied by one parent, part by the other. The substance of the parent molecules doesn't enter the crossover molecule. Currently, it's shown that when crossing over, there is always a real exchange of parts of two DNA molecules, and therefore the break and reunion hypothesis is most likely correct. This slide shows the crossing over in chromosome. The existence of the crossover allowed the school of Thomas Morgan in 1911 to 1940 to develop the principle of constructing genetic maps. The principle is based on the idea of the arrangement of genes along the length of the chromosome in a linear order. The genetic map of chromosomes is called the scheme of mutual arrangement of genes that are in the same linkage group. Determination of the coupling group is carried out by the hybridological method. That is when studying the results of crossing. Chromosomes are studied by cytological method using microscopy. By setting the linkage group, you can build genetic maps and specify the order of the genes. Let's assume the gene N and B belong to the same coupling group according to the results of crossing. The distance between them is 10 mogenets, 10% of the crossing. With further research, it was found that the C gene belong to the same groups. To establish its place in the coupling group, it is necessary to find out what percentage of crossing in it gives with of the two already known genes. For example, if in relation to the gene it gives 3% of the crossover, and in relation to the B gene 7%. Then it is located between the A and B genes. This slide shows the genetic map. You can see cytogenetic mapping and genetic high resolution mapping, physical mapping. If the number of crossover individuals is given as a percentage, the distance between the genes is equal to the sum of the percentage composition, where x is the distance between the genes mogenets, A and C the number of crossover individuals, and the total number of individuals. This slide shows the analysis of adhesion genes. You can see this scheme. So, gene linkage analysis is the test of joint or independent inheritance of diseases and genetic markers in families. Currently, genes for predisposition to many multifactorial diseases have been identified by analyzing the association of diseases with polymorphic genetic markers. The founders of the study of cytoplasmic heredity are Courant and Bauer. You can see portraits this scientist, Courant in left, for you and Bauer in right for you. The totality of hereditary inclinations of the cytoplasm is called a plasmon. Themselves marking plasmogene. The plasmon is part of the genome. The functions of plasmogene as genes located in the chromosomes bound to DNA. Plastids, mitochondria, and some other organoids have their own DNA. These cytoplasmic structures are capable of self-production. This is what the transmission of cytoplasmic inheritance is associated with. Plasmogene heterogeneous in nature. They can be divided into two groups. One, genes of DNA containing cell organelles, mitochondria, chloroplasts. Two, genes of infectious agents or cell symbionts, viruses, episomes, etc. Plasma gene of both groups are similar in their properties to nuclear genes and exercise genetic control over the synthesis of a number of important enzymes and consequently the development of some complex traits. They are capable of reduplication and point mutations that can be inherited. Features of inheritance through the cytoplasm. One, the number of cytoplasmic organoids containing their own DNA isn't contained, cannot be accurately accounted for. Two, in the fission of the organelles of the cytoplasm are distributed or maybe distributed between daughter cells univalently. Three, inheritance through the cytoplasm is not subject to the Mendelian laws of inheritance. 4. It occurs of the material line, since the cytoplasm of the zygote is formed mainly by the female germ cell. And 5. Cytoplasmic genes are controlled by nuclear genes. That's all. Thank you for your attention, dear students. Bye. Безумно весело, безумно интересно.